What's good, everybody? Welcome to a video on the physiology of a muscle contraction. Now, this is going to be another foundational video which will allow me to make cooler videos on how different compounds and training methods affect muscle tissue. Furthermore, if you haven't seen my muscle anatomy video, that would be helpful in understanding this one. With that being said, a muscle contraction actually starts in the nervous system. So it doesn't show it on the diagram, but there'll be a nerve running along this muscle fiber which will connect into this hole and to the T-tubule. So this nerve fiber will release a neurotransmitter called acetylcholine from its axon, which will depolarize the T-tubule, and thus it's kind of sending a signal through that depolarization. The T-tubules will then communicate to the sarcoplasmic reticulum to release calcium into the muscle fiber. Now this calcium then moves into the myofibril in order to communicate that it should contract. This next diagram zooms down into the thick and thin filaments of the myofibril. Now the thin filament is represented by this blue cord here, and the thick filament myosin is represented by the orange strip here with the kind of golf club sticking out of it. So we left off with calcium being released from the sarcoplasmic reticulum and traveling to the myofibrils. And so the calcium will then come and bind to this little blue ball here, which is actually a protein called troponin. Now troponin is responsible for moving this string of purple here called tropomyosin. And it moves tropomyosin so that myosin can bind to actin, because normally tropomyosin is blocking the ability of myosin to bind to actin. Now it's important to note that this myosin head is what it's referred to in the literature as, has an ADP and a phosphate group connected to it, which will make sense later on in the video. So we have calcium coming in, binding to troponin. Troponin moves tropomyosin out of the way. And now, once myosin binds to actin, the phosphate is released, which creates energy and a change in conformation that allows the myosin head here to pull on the actin. And this is referred to as a power stroke. This is when a muscle contraction is occurring. Once the contraction is complete, the ADP is released from myosin, and the myosin is now stuck in this contracted position until a new ATP molecule can come to bind to the myosin head. And when the ATP binds to this myosin head, it causes a change in conformation, meaning a change in shape of the myosin head so that it can be released from actin. Following this, the ATP is hydrolyzed. Now, hydrolyzation is a process that uses water to perform some kind of reaction. So in this case, the ATP molecule, which is adenosine with 3 phosphates, adenosine triphosphate, is cleaved so that one of the phosphates is removed, and it becomes ADP, adenosine diphosphate, and 1 phosphate group. Now, when the phosphate is cleaved, this allows for the creation of energy, and this energy is used to cock the myosin head back into position so that a new power stroke or muscle contraction can occur. So you can see from this process that the two rate-limiting molecules present are calcium and ATP. Thank you for watching the video. Please like, comment, subscribe. I'm always trying to get better, so if you have any feedback whatsoever, let me know. Have a good rest of your day.